Okay, we know. Um, Today I'll tell you about our work on organoids and trying to basically, Alan just told you about the end-of-life utricle. In our studies, what we're trying to do is actually produce a new utricle in a dish. Maybe. Okay, it's my turn now to be tormented by this thing. So how are we using stem cells? Kind of this virtuous cycle here is what we're most interested in, is being able to take cells from a patient, generate iPS cells through reprogramming. These cells can become any cell in the body, and it's our job to really figure out how to differentiate them, apply the signaling cues seen by the cells during development to allow them to and ideally generate multiple types of cells that we can then get to self-assemble into an organ-like structure or an organoid. And then the ways that researchers are using this, and we're doing this too, is for disease modeling purposes, drug testing, and then in some cases, organ replacement. And if you're thinking about the inner ear, we heard the name Albert Edge mentioned at one point. He's developed a nice model where you can take cochlear progenitor cells and grow them outside the body in kind of an organoid-like format. Unfortunately, you can't do that very well with human cells just because we don't have great access to either fetal human tissue or, as Alan mentioned, it's kind of a Herculean effort to get these adult specimens. So disease modeling, drug testing in the inner ear, those are things we're working on now. Organ replacement for the inner ear. I get this question a lot, like, are we going to take our organoids and plug them into somebody's ear? I don't think so. I've stopped thinking about that and dreaming about that, but maybe somebody can prove me wrong in the future. Okay, so why do we need these in vivo-like models? And you may have seen in the news, I think this was passed in December, there's this new FDA Modernization Act 2.0, which kind of lowered the barrier of drug testing so that you can essentially get a drug approved or a biologic approved without using any animal testing at all and just using cell-based therapies. And you might think that working on these models, I'm an evangelist and I really want organoids to replace everything. I don't. I think they're very complementary to mouse studies. And this passage kind of scares me because the models aren't really there yet. And it really puts a lot of pressure on us to make them higher fidelity compared to the native organs. So the work that we've been doing is kind of broadly focused on sensory organ development. And I like to put this slide up at the start of my talks just because it's a simple it shows you what we're trying to do. So we start out with this blank slate of pluripotent cells, and then we're trying to generate, in the case of a sensor, we want to generate epithelial cells. So like the sensory epithelia of the inner ear. In these three-dimensional cultures, they form these cysts with a lumen. And then we also have to think about the neurons and glia, like the wiring that connect a sensory epithelia to the brain. And then what's often forgotten is the mesenchymal populations, which are the support cells. And what we're learning in the stem cell field, generating all these different organ systems, is that each organ system has their own unique mesenchyme. And we know from studying the periodic mesenchyme in the ear that these cells really have an important role and function of the ear. So what we do primarily is trying to generate all of these components together in an aggregate of cells. But you can think about this, and this is something we're doing now, is generating each of the individual components separately and more efficiently, and then building the structure over time. So our primary contribution to the field has really been to generate these two sensory organs. So I'll focus primarily on our inner ear work, generating an innervated sensory epithelial structures, inner ear organoids. But at the end of the talk, if I get there, I'll tell you a little bit about our work on the skin, where we've been able to generate an innervated skin tissue organoid. Okay. And then we have several projects that have started up on Usher syndrome, a deafness blindness disorder. So recently we started growing retinas too. I can tell you that the protocols in the retinal field work quite well, and it was actually pretty easy to grow a retina. Okay. All right. So the key problem here, I don't have to go into a lot of background, but the key problem can be boiled down to this. Within the inner ear field, we don't really have a scalable system for investigating human inner ear cellular and molecular biology. And so that's what we're working towards. Our main challenge has been to mimic the inner ear development in its entirety due to the kind of problems with getting donor-derived tissues that Alan told you about. We've heard a little bit about inner ear development, but the key step at the beginning of the process is the formation of these two thickened patches of epithelium on either side of the embryo, the otic placodes that then invaginate, forming the otic vesicle, that then morphs into the organ we've been hearing about all day. And the key steps to recapitulating this process are really getting this otic placode and vesicle induction. So the work I did as a grad student and postdoc really focused on trying to better understand and apply the control mechanisms that cells see during development that induce these otic placodes, distilling down the wealth of information we had from the literature. Matt and Doris, your studies were really important for building a foundation of knowledge. And then applying those signaling cues, maybe, to shuttle the cells through these cell fate checkpoints to get to an otic progenitor-like state. Going from ESLs to generating ectoderm cells, surface ectoderm, pre-placode, and then otic progenitor cells. And I'll go into some detail on each one of these steps. So this is an overview of the process of organoid generation. We start out by dissociating the cells. We can do this with mouse or human stem cells. We form an aggregate of cells. Maybe something's missing here. There's a missing little detail here. We add extracellular matrix in the form of matrigel that kind of forms a basement membrane on the surface of the aggregate that stimulates an epithelium to form. And then we apply these different signaling pathways like BMP4, then FGF and a BMP inhibitor to induce surface ectoderm, pre-placode, otic progenitors, that then ultimately form placodes and develop these kind of off-budding vesicles that then mature and form sensory epithelia. Not depicted here, we also see co-development of sensory nerves that innervate the sensory epithelium. And I'll just show you a couple images of what this looks like with our mouse organoids, maybe. Wow, okay, yeah, very sensitive. Okay, so here's a nice comparison between one of our organoids at day 8 in a dish to a day 8 embryo, and you can see this kind of region of the embryo from which the otic placode develops, labeled by PAX8, is represented on the surface epithelium of the organoids. Much later, if we compare one of these fully formed inner ear organoids, you can see that they form these nice kind of cluster patches of myosin 7A, calretinin positive hair cells, similar to a vestibular organ. Also, you can appreciate there's cells around these epithelia, so we see co-development of a mesenchyme with the organoids. And then looking at the hair cells themselves, the sensory epithelia, we can see there's a layer of supporting cells, and then the vase-shaped hair cells with protruding hair bundles into the lumen of the organoid. So the key insight from this initial work was that we can partially direct the differentiation of these different lineages together and stimulate self-assembly of all these 3 main ingredients of an inner ear organ. We can now do this with human stem cells, and this is what this looks like just through bright field imaging. We start out with a fairly uniform aggregate of cells. By day 3, we see this outer epithelium representing kind of the early surface ectoderm. By day 6, this epithelium expands, kind of balloons up. There's an inner core of cells that I'll talk about in a little bit that actually emerge, form migratory cells that form a mesenchyme around the epithelium. And then we see these kind of protruding little buds by day 8 or so, representing otic placodes. Just to give you a little mechanistic insight, the kind of initial blast of BMP signaling is what's most important for kind of starting this process going and getting this nested structure. So applying BMP to the exterior of the aggregates induces surface ectoderm on the surface, not surprisingly. And then in the interior, we see that these cells generate neuroectoderm, which can form the central nervous system. But importantly, if we get the BMP signaling just right, we can get this combination of surface epithelium, future epidermis or placodes, and neural crest cells to emerge. And so the neural crest forms in the core and then emerges and forms a mesenchyme around the aggregate. And you can see varying the BMP concentrations we get, we can just look at the morphology of the epithelium and kind of determine the state of the differentiation. So without any BMP, this is a neural tube-like epithelium that's forming. It's much thicker than what you're having at higher BMP concentrations, which is more similar to the kind of lateral surface ectoderm. Okay, let's see. So this is what this looks like if we do a time lapse of the process. You can see that this initial epithelium forms. In the core, we see production of neuroectoderm and neural crest, and then ultimately this emerges and starts to migrate. These neural crest cells migrate out around the epithelium. And I want to highlight that we are primarily using reporter cell lines generated by the Allen Institute. They've got this beautiful catalog of reporter lines that have been really fantastic to use. This is a tight junction protein 1 GFP. Let's see. Okay. There we go. All right. Maybe. I hope this is started. It doesn't seem like it. Okay, these are not going. Oh, there we go. I'll skip that first one. So the first one you would see SOX2 positive placodes forming on the aggregates. Here are 3 representative organoids where you can see that after what this is 10 to 11 days, there are these protruding otic vesicle-like structures. So instead of invaginating like they do in vivo, they evaginate out into the culture medium, and then are eventually enveloped by that outer layer of neural crest mesenchymal cells. Oh, hey, don't worry about it. I'll move on. Okay. Okay, so much later in the process, the human development, not surprisingly, takes much longer than a mouse, reflecting the gestational timing, the differences between human and mouse. But if we look at the resulting organoids, you can see that they form these beautiful sensory epithelia, very vestibular-like, with hair cells, with hair bundles. We worked with Jeff Holt's group at Boston Children's with us to examine the electrophysiological properties of the hair cells, and we see similar potassium currents to nascent hair cells. And again, we see co-development of neurons that are innervating the sensory epithelium. So this is work that we published in 2019, and we've been kind of iteratively improving the process and developing the protocol over time. And kind of the general sense at the time of publication was that we can recapitulate most of what we saw in our mouse organoids. We can generate these vestibular-like sensory epithelia with like type 2-like hair cells. and neurons. And so what we've been working on, and I should mention that we take this approach where we're directing the differentiation, kind of controlling differentiation up front, and then we allow the tissue to self-assemble. Okay, wow. I feel like it's gotten angrier over time today, and it's really taken it out on me. So we've been working on a number of different cell lines, trying to make sure that this is reproducible across cell lines. And we've tested it out on, I think, 20 different cell lines now. We see subtle differences in how the cells respond to BMP. And that's really the critical part. If you're thinking about using this approach, please contact us. We have much better approaches for kind of quality controlling the early inductive events. We've also been looking at whether or not the hair cells and neurons are functional and forming a functional connection, getting into live cell imaging, and then working with collaborators at Boston Children's Hospital and elsewhere to see if we can model congenital deafness or gene therapies. Particularly, we have an emerging focus on Usher syndrome. And so a lot of the future goals for developing, further developing the system are to kind of garner more and more control of the induction. I mentioned that most of what we're getting is vestibular. So clearly working on that cochlear fate. As Doris told us, sonic's very important for that. I'm not going to talk about cochleogenesis today. Our friends in Indiana and the Hoshino lab have a nice preprint focused on that I think you should all check out. Very promising work on generating cochlear organoids. But we also have determined that there's kind of a need for basic knowledge about the system and then the inner ear development in general. So I'll tell you about our efforts to kind of single cell atlas the developing inner ear. And then I probably won't have much time to cover it, but we are also working on kind of more technical needs and innovations, trying to generate inner ear organoids on a chip. So one thing I want you to appreciate in this cross-section through an aggregate containing inner ear sensory epithelia is that there's a lot more biology going on here than just these inner ear epithelial structures. What we have come to appreciate is that we're getting more of a regional differentiation. And there are other accessory tissues that are forming along with the inner ear organoids. So we have a mesenchymal population that surrounds the epithelia, but then also we see cartilage development. And we think this is probably reminiscent of Meckel's cartilage or middle ear ossicles even that are forming or beginning to form. And then another major off target is development of skin tissue. So we see these cystic skin structures. And so two students in my lab, Wouter van der Valk, who's a visiting student from the Netherlands, and then Matt Steinhardt, who's an MD PhD candidate, just recently got his PhD. They had the question of kind of how diverse are our organoids, and can we really understand that diversity and better define it as a way of then resolving the cell lineages that are forming and then going back with a more data-driven approach to direct the differentiation and improve the process. What they did was they chose a cell line to do single cell RNA sequencing at different time points. For this particular study, which is in revision right now, they chose to use this desmoplakin GFP line from the Allen Institute, which seems to nicely label the apical surfaces of the developing epithelia. It labels the epidermis quite well, but also the apical surface of the otic vesicles, and it helps for identifying and quality controlling aggregates and showing that you can see nice vesicle development. And what we wanted to do was really define this early development of the tissue. That's really the black box that we can't get much insight into in normal human development. So we chose time points leading up to the formation of hair cells. So we sampled the cultures every 3 days, and then later on every 6 days in a second experiment, up to about day 36, which is about when we start to see hair cells developing. Okay. So here's where I'm going to start kind of glossing over the single cell data. But we've created this inner ear organoid developmental atlas, which we think really nicely captures the developmental diversity of the tissue and gives us nice insights into the different lineages that are forming. Just at a high level, we start out with pluripotent stem cells, and then we can see this immediate branching of surface epithelia forming, as well as neuroectodermal cell types forming neurons. And then within the surface ectoderm, we see diversification of different surface epithelial cell types like preplacoidal ectoderm, something called the otic epibranchial placode domain precursor cells, and then ultimately otic epithelium, which forms hair cells, as well as neurons. We can track the development of neurons branching off from these otic epithelia. And a lot of the visualization for this was done by a talented bioinformatician in my group, Daniel Osario. So as Alan mentioned, we also use the RNA velocity technique. We found that typical trajectory methods like monocle were kind of useless for this data because we think it's because we don't really need pseudo time because we have actual time built into the data sets. And the integrated data sets seem to be too complex for really showing us where the lineages were forming. So we used a combination of a priori knowledge and this RNA velocity to kind of piece together the different lineages that were forming. We're able to infer epithelial trajectories as well as non-otic neural trajectories, and then get some insights into how the mesenchymal cells were forming. One interesting bit of kind of surprise from this data set was that the neuron situation is a bit of a mess. If you look at the developing cranial region, in which the inner ear forms, there are a number of different nerves that are developing, like the 3 epibranchial placodes form different ganglion neurons, as well as the otic ganglion, trigeminal ganglion, and then the hindbrain adjacent to these structures. And we see signatures for each of those showing up within our organoids. So if somebody wants to use this model for looking specifically at these different neuronal populations or specifically at otic neurons, then they really need to factor this into their analysis. Hang on. To summarize our key findings from this, we have a number of insights that really clarifies this initial split of the development of surface and neural development. I didn't talk about it, but there's actually a signature for amnion-like cells within this, which we didn't appreciate, probably driving this ballooning of the structure over time. We see both anterior and posterior epithelia, and so that's giving us hints about how we could improve the efficiency of induction. Surprisingly, we also see periotic mesenchyme forming, which I'll show you some images of in a bit. And we provide some guidance on kind of how you would use this, this map that we've generated. And this is kind of not showing up very well, but one way we've demonstrated how you could use this atlas is if you're starting out using the organoid technique, you can use this as a benchmark for comparison. And so here we've used a software called Symphony to map cells derived from our collaborative group in Zurich, led by Marta Rossio. So they've taken a different cell line, undergone or performed this differentiation, generated single cells at the same time points as us, and then we can map their data onto our data and get a sense of how well they've reproduced the inductive process. And I think just by comparing the pattern of cells, you can appreciate it's worked quite well. Okay. Yikes. Okay. Okay, great. But what about more mature time points? I think people are most interested in kind of the later time of development of the sensory epithelium. So in addition to that first study, we have another study coming out soon where we've created an integrated atlas of both human fetal, adult, using, I think, one of these adult vestibular schwannoma derived data sets and organoid inner ear hair cells at later time points, day 75 and 110 of the process. And this was led by Wouter in the Netherlands. And so he's created it's not going, okay. He's created an integrated atlas, and importantly, this atlas contains cochlear epithelium from the fetal specimens, which we think is particularly illuminating for comparing to our organoid tissue, which and thinking about or identifying potentially new ways of generating cochlear organoid tissue. Okay, and this work is probably going to come out in Cell Reports next month. And it's also going to be available on the GEAR very soon. We're working with Rona's team and the GEAR team on this. So I'm not going to talk about hair cells. I figured we'd beat hair cells to death by this point. So a couple key insights here was that we do see periodic mesenchyme forming surrounding the sensory epithelia, which is interesting to see. That's labeled by this very specific marker, otoraptin, OTOR, as well as PAU3F4 is labeling these periodic mesenchyme cells. Another shocker was that we see endothelial cell development within our organoids. And so you can see stained by CD31 endothelium coursing through our aggregate of cells and particularly adjacent to the otic epithelium. I'm not including it here, but we've done quite a bit of cell-to-cell communication analysis using cell chat. And one of the really interesting interactions that popped out of that just repeatedly through different pathways was hair cells and endothelial cells. And seeing Dan's data with the Mueller cells interacting with the endothelial cells makes me think that there's some there, and we should look harder at that. Okay, so the key findings from both of these studies have really been to broaden our understanding of the different lineages that are forming. Clearly, we're getting a broader induction than we originally thought. I didn't dig into the signaling mechanisms, but we do have a wealth of insights into kind of what signaling mechanisms we could tweak to get maybe a more efficient induction of the otic pathways. And then we were able to identify a greater number of otic epithelial and mesenchymal cell types. I didn't include it, but we do see evidence of kind of nascent type 1 hair cells. I don't want to say they're frank type 1 hair cells with Jenny in the room, but I think that they are nascent type 1s. We do see calical Indian-like structures. So one way we're using this data set, and actually I know it's kind of boring, but there's a beautiful supplementary table 1 in this manuscript where Wouter's gone through and shown and identified all of the 150 or so hereditary hearing loss genes, which ones are expressed within the organoids, how confident we are in that expression, have we validated them? And so we're using that to prioritize targets for disease modeling. And I've just labeled a handful of those here, like stereocylin, and then a number of usher genes that we're looking at. And that's led by a postdoc in the group, Jingwan Zhang, and a graduate student, Karl Koehler. And this is work that, or this work is really kind of bolstering our precision medicine team that we've developed with Marley Kane in the clinic and Elliot Shearer also on the clinical side, helping us with genetic diagnosis, looking at molecular mechanisms, and then our collaborators Jeffrey Holt and Gwen Jeliak on the basic research side to create kind of a pipeline for precision medicine, where patients come in the clinic, like an Usher's patient, and we could use CRISPR-Cas9 to edit our favorite cell line with their particular mutation, and then design a gene therapy to test out in the organoids. Okay. Now, I have a lot more, but I'm going to stop there. I wish I could leap to my acknowledgement slides. So I'd be happy to take any questions, and I'd be happy to show you the bonus material if you're interested. Yeah.
We have some questions from the chat.
Sure. So this is from the online audience. Justine asked whether you see different population neural crest cells like glial cells versus melanoblasts.
What's that?
Glial versus
Oh,
melanoblasts.
yeah. So I didn't get into the glia, but we do see a pretty sizable population of probably Schwann cell precursor cells and then non-myelinating Schwann cells. And we do see interaction between those Schwann cell populations and the neurons that are forming around the otic vesicles.
We do have another question. Can we use the organoids to study conditions like tinnitus, hyperacusis, or vestibular disorders?
Yeah, so that's a great question. I think that the lowest hanging fruit is going to be looking at like monogenic disorders. So one that we're really keen to look at within the organoids, where we really think we're going to see a clear phenotype, is looking at myosin 7A related mutations. And that's something we're going to be exploring in the very near future. Yep. Oh, Jaime?
It's SOX8 somewhere involved in the beginning of this. There was this paper in PNAS about it inducing otic neuroepithelium. I just don't know if you.
SOX8. Yeah, I think I know of the paper that you're referring to. I think we've looked at that. I think it's expressed. I don't know if.
I just wonder whether inducing it or inhibiting it or whatever. You haven't done anything with it.
Yeah, we haven't done anything with SOX8. Yeah. Oh. Doris.
Hi, that was really nice. I was wondering for all the people that try to make organoids, it's probably is not your focus, like to put neural tube in there.
Yeah.
And I don't know if that was even something within your scope of interest, and if whether it's doable.
Yeah. So what we've discovered through our single cell analysis is that there's probably a variable number or amount of neural tube that's co-developing with the inner ear organoids. So it's very possible that there's a little bit of hindbrain developing with, or brainstem developing with the inner ear organoids. We haven't looked at whether or not there's a connection between the neurons and that a little bit, but I mean, ultimately we'd want to remove that. And so some of the stuff I didn't get to is focused on better controlling the development and the spatial patterning of the organoids. Trying to remove some of those off targets physically by sorting out cells and then putting them into a microfluidic chip-like platform.
I see.
Yeah.
So you can control.
Yeah.
Okay.
And there are better protocols now for generating hindbrain or brainstem-like organoids. The problem is we don't really know that much about how the brainstem is patterned during early development. So how do we get to those different nuclei, much less the cochlear nucleus and like specific nuclei that we're interested in.
Right. Yeah, it's crazy hard, I bet.
Yeah.
I mean, we're running out of time. I mean,
Yeah.
Like, you're gonna